Hello, I'm Raphael.
I'm Shuming, Xu, and I'm Liang, Xiang.
We're Group 8, and we're working on decoding neural signals into intended movement. So to give a little background, approximately 3.4 million people in the United States suffer from paralysis. This can be due to neurological injury or disease, which can lead to an inability to care for oneself, communicate with others, and live a normal life. Next slide. So a little rationale behind why to use a brain-machine interface. The goal is to bypass the intended neurons by measuring directly from the brain, reading the neural signals, decoding the intended movement, and then coding the prosthetic to move as you would have intended. Next slide. So a little bit about the normal physiology. Initially, you have the subcortical areas. They give the initial intent to move. Then the association cortex, which includes the cortical areas located between the visual, auditory, and somatosensory cortices. Those create a movement design. And then that goes to the basal ganglia and the cerebellum, which refine that movement plan. So the basal ganglia is responsible for processing movement and sequence planning, while the cerebellum helps with timing and coordination. Then that goes to the thalamus, which relays it to the motor cortex. And the motor cortex sends that information down to the motor units to actually execute this movement plan. So a little bit about people who have paralysis for a long time. They have the potential for cortical reorganization. That's where, due to a lack of somatosensory feedback, their motor cortex reorganizes into activating different areas because the paralysis no longer allows them to activate those previous areas. So in this example here, we have the rat is intact with an intact spine, and then in figure B, you can see that after a spinal cord injury between the C2 and C3 vertebra, there's no longer activity for the activation for the hind limb or the trunk, and instead you have the whisker overtake those previous areas. And you can see a similar occurrence with another rat in with a spinal cord injury between the C4 and C5 vertebrae. This poses a problem because remapping could remove areas that would normally be used for decoding in a brain-machine interface, and that would make decoding very difficult. But there's also potential for remapping when training with using a BMI, which can improve results for decoding.
Okay, so next I'm going to talk about the engineering implementation of the BMI system. So this diagram basically shows the framework of all the parts of system. First, there's a neural signal. The electrode records neural signals from the brain, and then there's certain extraction or decoding algorithms, which translates the neural signals into the control signals of the effector. And the effector, such as a robotic limb, will move according to these control signals. And then finally, there's a feedback being sent to the subject. For the electro recording parts, I think we have covered this a lot in class. The four major modalities is EEG, echo, local field potential, and single unit action potentials. And for the decoding algorithms, it translates the neural signals into movement commands of the robotic limb. So before we know how to translate these signals, let's first talk about how the movement directions is encoded in the motor cortex. So there's a very basic findings by Geo Joblos and his colleagues in 1980s. The main conclusion of their experiment is that the actual so each neuron has a preferred moving directions, where it fares at the maximum rate. And the actual moving direction in the physical space is just the preferred moving directions of each neuron, some of them weighted by their varying rates. So based on these findings, there's the algorithm called a population vector method. It's basically just a linear regression method that maps the varying rates of vectors into the hand motion vectors. And this algorithm, although quite simple and naive, but it has proved to be very efficient in many application scenarios. And after that, there's an effector which can be either a cursor on the computer screen or an actual physical artificial limb. And the feedback is just about the training stuff. Like talked before, the subject will learn how to use the device through training over time, and the performance can improve significantly. And here are some short videos on an experiment being done by a human with tetraplegia and this task is about how to control the patient try to use the neural signals to control the cursor to a certain target on the computer screen. And you can see this kind of task is quite easy because the output is just a XY coordinate. And the second task is the person try to control a robotic arm through his neural signals, and it tries to move one thing from one place to another. And you can this kind of task is more challenging because there are more degrees of freedom, and it needs more sophisticated control of the device. And there are more interesting videos on this from this paper. You can click the link if you are interested. And finally, the challenges and future directions of BMS system is first the robot is listed here. First is the robust long-term recording techniques, which is a common issue for almost all the engineering devices. And second, we need real-time and high-performance decoding algorithms, which can be run online. And third, we need so far, most devices only use visual feedback, but actually somatosensory feedback is also important. It will allow more efficient training and make the subject feel like the artificial limb is part of his or her body. And what we talk about are just most of them are just prototype in the labs, but in order to be used in clinical applications, they should be portable and should be able to wirelessly transmit neural signals into the robotic arms. So that's all for the review parts.
Well, next, I'm going to present our MATLAB simulation. The general idea is that we should simulate the data analysis process when we decode neural signals into intended movement of limbs. The data consists of two different parts. The first part is the ECOG data, and the second part is the corresponding finger movement data. And next slide, please. First, we'll have an overview of the data. For the ECOG data, it is collected with an 8x8 electrode arrangement with two reference channels. The sampling rate is 1 kilohertz, and we have 300 seconds of train data and 100 seconds of test data. Next slide, please. The finger movement data is collected using a data glove. And each finger is ordered to move 30 times during the experiment, and it's sampled at 25 hertz and then upsampled to 1 kilohertz. Next, please. Then the algorithm is as such. First, we do the labeling of the finger movement. For the kth finger, we label -1 as no movement and 1 as movement, getting the label vector of the length of time. And we add an additional dimension, y6. It is 1 when none of the fingers move and -1 otherwise. So there is a total of 6 label vectors. Next, please. And the second step is channel selection. It is to reduce the computational effort and boost the overall performance. First, we divide the original data into bins of 200 samples. And for every bin, we apply an autoregressive model and take the first AR coefficient as a feature. Then we spline interpolation to get values of all the time points, getting a feature vector X, which is time by channel. Next, please. And for every finger, we get a linear regression model in which we have the linear coefficients, the length of channels. And the relevance of every channel will be the sum of the absolute value of the linear coefficients. And the M channels with the largest S will be selected. M is a hyperparameter which needs tuning. So this is the whole channel selection part. And the next step is feature extraction. The basic procedure is the same, but we introduce 4 more features from 2 shifted signals. One is shifted left by 40 milliseconds and one is shifted right by 40 milliseconds. It is to take into account the delay between brain activity and finger movements. So we have a total of 6 features. And the next slide, please. Then in the modeling step, we choose ridge regression since the movements of different fingers are somewhat related. The ridge regression is a modified version of least squares method in which a regularization component is added. In the equation, lambda is a hyperparameter that needs tuning, and W is the modeled weight. Next. And for the performance evaluation, the competition we get our data from used the correlation coefficients of the actual movement and predicted movements as a criteria. This is a table of the performance of all the submissions, and our performance is on the right. The mean correlation coefficients of our implementation is 0.33, which is decent among all the entries. And on above, we can see the individual correlation coefficients for every finger. We can see that it is pretty good for thumb and index finger, but for middle finger, ring finger, and little finger, it's kind of low. I think it is because that the movement of the 3 fingers are kind of related. It is almost impossible that you do not move one of the other fingers when you move one of these. So that's kind of why we get these results. Next, please. And this is a figure we get on the prediction side. We use a simple threshold method to predict the finger movement. The figure above is the predicted movement, and the figure below is the actual movement. I think the result is quite decent, but if we are given more time and more hyperparameter tuning, I think we can get a better result. And that's our MATLAB simulation.
And thank you for your listening. That's all.